Good morning, afternoon, everybody. I hope that you guys enjoyed your break. I hope that you're having a good Tuesday. We got they will be entered before the end of the day, before 5 p.m. I will say that. And then I'll release I'm going to then adjust or duplicate the scores. So then if they are higher than your exam two score, those will be replaced. Um, okay, so I will take care of that. Then I got a private message question. The final for next week is at the same day and time as another final that I have. Can I take it a different day? Um, okay, so here's the final exam is scheduled. Let me share my screen. Your final exam is so if you think that your final is scheduled for the exact same time as another one, I would make sure to double check because um the registrar's office tends to make sure that there are no finals administered at the same time. Okay. So I'm going to talk about the final for a minute, then I'm going to talk about the project, and then I'm going to get into today's content about lipid metabolism. So I've gotten some private message questions. I want to go ahead and address those in just a moment. So your final is next week. That link that I just sent takes you to this page. Click Fall 2025. Our class meets on Tuesdays and Thursdays. So if you look in the far left column, class meeting days, you'll see Tuesday, Thursday. Our class meets at 12:30 from 12:30 to 1:50. Our final exam is therefore scheduled on Tuesday, December 9th at 2025 until at 11 a.m. until 1:30 p.m. Now, I am going to open up the final exam testing window. I'm going to make that available from 7:30 a.m. until 8 p.m. So you'll have basically all day to take the final exam for this class. In the event that you have three exams, I think the university makes accommodations to take exams on a different date. If you have three exams on the same date, if you have that, or if you have some overwhelming circumstances, please let me know, and we can figure out a good time for you to take the exam. Other than that, though, you will have the bulk of Tuesday, December 9th to take the exam. Okay, so the final exam is going to be, and now I'm going to switch and I'm going to give general details about the final exam. And then I'll talk more about that on Thursday. Okay, so final exam window will be Tuesday, 1 week from today, 12/9, 7:30 a.m. until 8 p.m. 2 hours, 80 questions. Um, in terms of what's going to be on there, everything. I will send out, I'm finalizing my draft of the final today, and I will send out guidelines and suggestions of what to look at, what to review. Will there be stuff about lipids, membrane transport, lipid metabolism, stuff that we're covering today? Yes. Is there new content that we're going to cover on Thursday? There will be a little bit. Will that be on the exam? Yes. Will there be a disproportionate amount of stuff from the past few days? So last Tuesday, the asynchronous stuff that I sent you, that module that I sent you, and today and Thursday, will there be like 50 questions on that? No, there will not. If I had to make an approximation, you're looking at maybe 12 to 13 questions from the most recent stuff that you have not been tested over. So I'll put that as membrane transport. I'll say membrane export, comma, lipid metabolism, and biosignaling. Could everyone give me a one in the chat if you understand that this stuff that I'm underlining is stuff that you have not been tested over? Okay. Probably 12 questions. 12, I'll say 12 to 14 to give myself a little bit of latitude. 12 to 14 questions on these. The remaining, what would that be? 66 questions will be over stuff that we've covered throughout the entire semester. Could everyone give me a seven in the chat if you understand kind of those two details of the distribution? Okay. I will go over more detail on that on Thursday, and I will be getting you more review materials before our class on Thursday, including a practice exam. Okay, so I wanted to talk about the final exam, then I wanted to talk about the project. So now I want to discuss the project and the details about the project. Okay, so one of the big questions that I've gotten, and I fully understand why you asked this question, is the project extra credit? The answer to that is no, it is not extra credit. And for that, you might think, well, how is it not extra credit if it's optional? So I'm going to say, is it extra credit? No. Is it optional? Yes. What I mean by that is, let's say that it, so I'm going to give you a hypothetical student. Let's say that they do the project, or yeah, so they do the project, and let's say on the project they get 10 out of 10 on that project. And let's say that without doing the project, so not doing the project. Okay, so this hypothetical student, let's say that they come into this with, let's say that they have an 85 average in the class. And this is after the final, after everything has been accounted for, they have an 85 in the class. And so if they do not do the project, they'll end up with a B in the class. Could you give me a 9 in the chat if you follow that? Okay. Now, in the event that they do the project, what I'm going to do is I'm going to take that 85, that average that they have in the class, and I'm going to multiply that by 0.9. The reason that I'm going to do that is because this 10 out of 10 will be that last 10% of their class grade. So I will take the 85, the average that they have with everything else, multiply it by 0.9, and then I will add to that 100 times 0.1. So if they do the project, their grade would end up being, let's see, 85 times 0.9. That would end their semester with an 86.5. Could you give me a 5 in the chat if you understand the math and why I did the 0.9 and 0.1? So this is not extra credit because if it was extra credit and it was worth 10 points or something like that, that would propel that 85 all the way up to a 95. And that's not the case. Will this change someone's grade in the positive? Yes. Has the potential to do that. Okay, that's a great question. So the project can only help your grade. I think that's a reasonable takeaway. The reason that's a reasonable takeaway is that is because what I'm going to do is I think that the if you're submitting something, there's pretty clear, or to me, I think that there are clear directions as to what you need to submit. Um, you need to, and so the chance that you would get anything but a 10 out of 10, I would say is relatively low. Great question. Are we going to lose points if we don't choose to do it? If you elect to not do it? Well, no, because if you're the student who rolls in with an 85 and you come in and you elect to not do the project, your grade is going to stay at an 85. Now I got a follow-up question. So is the grade we have on Canvas our final grade as of now, or do I still have to input some grades? I still have to input some grades. I will have your grade up to date by the end of the workday on Friday. I'm going to try to do it sooner than that, but that's what I'm going to plan on. So yes, there are things that still need to be input, and there are things that will ultimately be dropped. And of course, the calculations with the exam grades. The project is due 12/12, so a week from Friday at 11:59 p.m. Are we going to get our final exam grades in before the project is due? Yes. So if you want to wait until next week, say Wednesday or something like that, and say, I guess I'll do it, that can shift my grade up to a point and a half or so, then go for it. The assignments on Canvas that are due on the 4th are locked. Are those a part of the project? The assignments that are locked, due on the 4th. Okay, I got a private message question. Well, let me adjust that. If they're locked, then I need to open them up and make sure that everyone can actually do them. It's part of the module, yeah. Let each things complete. Complete. Yeah, that makes more sense than omelet. Lock it. Okay. It's, okay. I don't know. So there is something that you have to mark as done. Dang it, that's not what I meant. So that means you at the bottom of the screen, which I will share my screen in just a moment, you have to literally click mark to show that it's done. Um, you as a student. Okay, just a second. So you need to click mark as done. I'm not sure if you attempted that and that was unsuccessful or what. And that will take you on to the next thing. Has anyone encountered that and found that to okay. It looks like yeah. I understand how that's confusing. It shouldn't be locked though. You should be able to navigate through that without an issue. But you do have to click mark as done. So if you have not done that, then go ahead and do that in order to complete it. Now, in terms of the things that are being graded, that's really just the quizzes, which those are counted as homework assignments. Okay. Now, if you have any questions about the project, um, and how it would impact your grade, please let me know. I'm glad to answer that. We can meet on Zoom. I can plug in hypothetical grades for you. So we've talked about the final exam, the details about the final exam. This is going to be posted on Canvas as the recordings typically are. We talked about the project, and now we're going to talk about some new content for today. Now, lots of what we're talking about today is going to be context. I'm going to highlight some of the very important parts of it and what I think, or what I really want you to kind of focus in on and remember. Now, with that in mind, we've talked about lipids lately. We've talked about the roles that lipids play. And now we're going to talk about that energy role or that energy storage role that lipids play. Now, if you think about what we've discussed this semester in terms of metabolism, does anyone remember the name of the pathway for breaking down sugars? If you do, add it to the chat. I'll give you a hint. I'm looking for a word that starts with a G. Glycolysis. Perfect. Now, if you remember glycolysis, we took a 6-carbon molecule, a 6-carbon glucose molecule, and we broke it down. We collected a couple of different products. We collected ATP. Great. That's our energy currency. We collected that adenosine triphosphate. We also ended up with 2 3-carbon molecules of pyruvate. Does anyone remember the other major product of glycolysis? And I'm looking for an acronym that starts with an N. NADH. Perfect. So we also talked about NADH. So in terms of glycolysis, you input 6 carbons in the form of glucose, you get ATP, you get NADH, and you get pyruvate. Now, pyruvate is a molecule that I want you to visualize as, okay, it's 3 carbons. Well, what you do with that molecule is you want to break that down as much as possible. You want to break that 3-carbon molecule and transfer the energy from those 3 carbons, which are shown here, into NADH, but you want to essentially break that 3-carbon molecule down to CO2. So if you ever hear the term cellular respiration, well, you're taking a fuel source, glucose, for example, and breaking it down into CO2. I always think of cellular respiration as a good term to know because if I'm inhaling O2 and then I'm exhaling CO2. So that's my respiration, my breathing, input of O2, output of CO2. Now, what we're doing is we're taking the 3 carbons from pyruvate and we're generating NADH. Now, this pathway here is known as the citric acid cycle, also known as the Krebs cycle, also known as the TCA cycle. TCA stands for tricarboxylic acid. Now, if you elect to take metabolism, you'll get into this pathway quite a bit more. The important takeaways for this pathway, however, are this NADH. That NADH is one of the major products of the citric acid cycle. So we get it here, we get it here, we get it here, and then we get another molecule, FADH2, which is similar to NADH. Now, the important part about FADH2 and NADH is they are electron carriers. Could anyone tell me, oh, I got a private message question about the final exam, and I'm sorry to interrupt myself on this, but do we get a note card for the final exam? Yes, you can use a 3x5 note card or index card for your final exam. You can use just one though. Okay, so the idea is that you've kind of pared down what you're bringing with you to the exam. Okay, so now back to electron carriers. NADH and FADH2. These 2 compounds are electron carriers. Could you tell me If something is electron carrier, what do you think that it does with those electrons? Do you think that it's going to grab onto electrons and hold them indefinitely? Or do you think that maybe it would do something with those electrons? Maybe do a type of reaction? Um, could you tell me reduces? That's what I'm looking for. And I know that was kind of a clunky question, but that's what I'm looking for. Things that are electron carriers can participate in redox reactions. Now, if you think about a redox reaction, it might not be super exciting when you're just talking about, oh, well, NADH becomes NAD plus. Ooh, might not be too fascinating, but let's think of some other redox reactions. If you took, let's say, magnesium and added it to a strong acid like HCl, um, what's going to happen to that reaction? Just give maybe some ideas either of the products or maybe visually, what are you going to see? Well, one product that you're going to get is MgCl2. Ooh, neat. What's another product you're going to get from this reaction? I'll give you a hint. If you're standing over this reaction, it might burn your nostrils. It's a gas. H2. Perfect. Now, H2 is a gas that's produced, but also that's a flammable gas. So in a redox reaction, there's a lot of energy that's being transferred. Now, in addition to that, visually, if you're watching this reaction, it's going to be kind of violent. There's going to be bubbles and gas and everything forming. So it's a little bit exciting. So redox reactions do involve the transfer of a lot of energy. And that's what I want you to take away from the citric acid cycle. You're getting these molecules that will ultimately harbor a lot of energy. So essentially what we're doing with the citric acid cycle, also known as the TCA cycle, also known as the Krebs cycle, is you're taking those three carbons of pyruvate and yeah, you're getting rid of CO2, but maybe more importantly, you're generating NADH and FADH2. Could you give me a 9 in the chat if you, okay, I follow that. We're getting rid of carbons. Okay, now the important part of all of this, and actually, let me think. How many carbons does, I'm going to write a code on the screen. You tell me how many carbons does this molecule have? How many carbons does that molecule have? Great. Now I'm going to talk about a fatty acid. I'm going to label it FA, and I'm going to say C16 colon 2. How many carbons does that have? Okay, great. Now with this 6-carbon glucose molecule, we're going to get some NADH. Cool. Do you think that you're going to get more NADH from a 16 carbon long fatty acid? Give me a Y if you think that this will get more than glucose, than a 6-carbon glucose molecule. Okay, great. You're right. Now, with that in mind, I want you to be thinking about that as kind of our primer, and as like background information that's going to be valuable. Okay. Now, could anyone tell me what organelle within a eukaryotic cell does the citric acid cycle take place? That's right, the mitochondria, the powerhouse of the cell. And my wife is an attorney by training, but whenever I say mitochondria, she took high school biology. She took a freshman biology class as a college student, and she still kind of has the motor, the what is it, the muscle memory that when I say mitochondria, she says powerhouse of the cell. So I like to imagine that you guys are sitting in your homes. I said mitochondria, and you went powerhouse of the cell. Okay, now with that said. Here's the mitochondria, Or here's the membranes of the mitochondria. It's got two membranes. These two membranes are pretty darn important. I like it. Private message that said Bill Nye solidified that. I think that's true. Yeah. So the two membranes of the mitochondria basically serve as a really good model for separating stuff that's in the cytosol versus the interior of the mitochondria. So our two membranes, we've got the outer membrane and the inner membrane. Now, with that in mind, you've got space between those. So your outer membrane, well, it's got outer face, inner face, okay. Inner membrane, outer face, inner face, okay. What you should take away from the outer membrane of the cytosol is that it is freely permeable to molecules that are up to 10,000 Daltons. So 10,000 Daltons. I've mentioned this before. Does anyone remember approximately how large one amino acid is in Daltons? One amino acid is approximately 110 Daltons. I think I might have said anywhere from Daltons. Anywhere from like 107 to 114. So what that means is that this outer membrane, if you have a protein that's like 75 amino acids, do you think that can move in and out of the outer membrane of the mitochondria? Give me a Y or N. And I know that this is some mental math here. 75 times 110, 7,500, give or take. Well, give. It's going to be a little larger than that. But yeah, small proteins. That looks like it says JMALL. That's just my poor handwriting. Small proteins can move in and out of that outer membrane. The inner membrane, however, is very selective. The inner membrane is the one that matters. The only things that are permeable to the inner membrane is water, CO2, and O2. Everything else has to be moved across with the help of a protein. So that means a membrane protein has to be embedded in here in order to take something from this what's known as intermembrane space. So that space between the two membranes. So there must be a protein that moves those things from that intermembrane space into the mitochondria. You give me a 6 if you follow. Okay. So let's say hypothetically that we've got pyruvate. If you think about 75 amino acids, or sorry, let me rephrase that. Yeah, 75 amino, I'll keep that. 75 amino acid molecule versus pyruvate. Is pyruvate smaller? Absolutely. Pyruvate's only 3 carbons. Could pyruvate move across the outer membrane? Give me a Y or N in the chat. Yes. Could pyruvate move across the inner membrane freely? No, it can't. It has to have a transport protein, a protein that moves it across the membrane. Okay. If you understand that, that's a really great place to start in. Now, to understand this, though, and we're working our way to lipid metabolism. I just want to give you the context for what are we doing with this pyruvate? How does it get into the mitochondria? So this, if we start up here, all of this in the upper left-hand corner, that's glycolysis. From one glucose molecule, we got 2 pyruvates. This black border here, that is our inner membrane. Just kind of visualize that as our inner membrane. We can move 2 protons into the mitochondria. And whenever those 2 protons are moved into the mitochondria, they can enter the TCA cycle, which is kind of highlighted right here. I'm circling acetyl-CoA and TCA cycle just because that's the takeaway of it all. And from that, we're going to get, just so we can keep the math straight, from 2 pyruvates, we'll be able to get 8 NADH. Cool. 8 NADH and 2 FADH2. Awesome. Now, These electron carriers, these two molecules that have kind of excess electrons, what they are going to do is enter this thing known as the electron transport chain. Could you give me a one in the chat if you've ever heard of the electron transport chain, or any other number if you've never heard of that? Let me just say outside of this class. If you've heard about it and you have a good understanding of it, awesome. That's going to help you here. It's also going to help you in metabolism. We're not going to go super deep into the electron transport chain, but the important takeaway is that these electrons from NADH and FADH2 are transferred onto O2 to make H2O, and that whole process is going to propel protons out of the membrane. And those protons that I'm putting a star by, where do they get caught? They get caught right here. Now, could they move across the outer membrane? They can. They can. They absolutely can. They are super small. They're protons, but they can't move across the inner membrane freely. Despite a single proton being smaller than a water molecule, an O2, or a CO2 molecule, they can't freely move across. Okay, so these protons, they kind of get locally stuck in one location. And then we use this right here, a protein complex known as ATP synthase, that is going to move protons from one side in the inner membrane space to the interior of the mitochondria. And what is that going to do? So this proton, or these protons, I'll say protons, multiple protons, will move across. And when that is happening, an ADP molecule and an inorganic phosphate molecule will be combined to make an ATP molecule. That is a process known as oxidative phosphorylation. Okay, so give me on a scale of 1 to 10, or 1 to 3, 1 being I'm good, 3 being I've totally blanked on this. How do you feel about this section right here? Okay, now what about, I'm going to switch colors so I can distinguish a different spot. This part right here, 1 to 3, how do you feel about that? Keep in mind, have I gone into the specific details of the citric acid cycle? No, I haven't. Am I going to? No, I'm not. Why? Because that's not a part of this class. Okay, so I'm seeing 1s and 2s. Okay, now I'm going to highlight this whole thing right here. How do you feel about that? 1 to 3. In summary, what you could say is I'm going to say 1, 2, and 3. And I'm going to give you a simple statement. 1, glucose makes pyruvate. 2, pyruvate makes NADH plus FADH2. And 3, FADH2, or I'm going to rephrase that to E carriers move protons out, ATP synth moves protons in and makes ATP. So those, if you were to sum those different blocks up, I think that would be a good way of looking at it. Okay, so this is all a sugar-centric approach to it. So this is like, okay, what is glucose doing in order for this to happen? How is glucose contributing to the generation of more ATP? Now, we talked about glycolysis where, oh, we get 2 ATP. Cool. But if you look at this, And it's not showing all of the numbers, but glucose makes 2 ATP. Well, NADH and FADH2 make a whole lot more ATP. And we're going to get into why that is today. Now, with that in mind, I want you to switch gears for a minute. We're now going to talk about fatty acids and lipids. Okay, fatty acids are a major part of membranes. They are your membranes, basically. You've got proteins in membranes as well, but lipids, in addition to serving as major parts of membranes, they're energy stores. And if you remember, we talked about polar and non-polar lipids. Your non-polar lipids are primarily your energy stores. Now, if you look at this point right here, this tells us that for 1 gram, a single gram of a lipid, and you can say a fatty acid, whatever, you're going to get approximately 38 kilojoules of energy. The context of that might be a little bit mixed up, but more than anything else, that's 2 times the energy that you get from carbohydrates. Now, to translate this, I know that if you talk to like a dietitian, you'll be told 1 gram of carbs is approximately, if I remember correctly, about 4 calories. Anyone who remembers that better, please don't hesitate to correct me. 1 gram of fats. Could anyone tell me approximately what this is? Uh, yep, it's about 9 calories. That's why people will, whatever, talking about, well, what are your fitness goals or what are you trying to eat? How many calories do you want to ingest in a day? They might say, well, I want to reduce the amount of fat that I'm consuming because, well, 1 gram of carbs, well, that's a lot less. 1 gram of fat is approximately 9 calories. It's nearly 2 times that. Now, 30 to 60% of dietary calories or lipids amount to approximately 30 to 60% of most folks' dietary calories. Diet nutritional guidelines are going to vary, and of course, speak with your dietitian for whatever your goals might be. Um, or if you have any sort of dietary related goals, or if you have some sort of diet where it's like, oh, well, you know, you don't need to necessarily worry about the calories that are intake or that are taken in, but instead you need to think about these other components. Okay. Now, approximately 90% of dietary lipids or your dietary fats are in the form of triacylglycerols. That's where we had our glycerol backbone. And then we esterified them to get something like this, R1, R2, where we have those long alkyl chains, R3. Now, these are all just sort of examples where we have palmitic and stearic acid, which are saturated fats, and then these right here, which are unsaturated, and these three right here are polyunsaturated fatty acids. 18:2 is a delta 9, delta 12 fatty acid, which is also a omega-6 fatty acid. Now, here's an example of a triacylglycerol. Okay, cool, lots of carbons. I got to be honest, whenever I think of fats and I think of diet, I don't necessarily think, ooh, carbons. Instead, what I typically think about is food. I think about chocolate. I think about chicken wings. I think about butter. I think about olive oil. Those are the things that I think about. And so the connection from that to this is a little bit tough to like put together so abruptly. Now, over the next chunk of slides, and I think it might be, let me check. Let's see, it's next like 10 or 12 slides. This is going to be kind of a crash course of what happens whenever this food is ingested or a fat is ingested. Okay, so let's say that uh, Here's your overview of what's happening. Now, I've broken this down into individual steps, into individual components. Okay, so what we have here is your liver, and we've got dietary fat. Now, when dietary fat goes into the stomach, it triggers the production of bile acids, also known as bile salts. Bile acids are produced in the liver and they're stored in your gallbladder. Okay. So you might be thinking, what on earth is a bile acid? What is a bile salt? Huh? Before we get to that, I want you to remember what we talked about with respect to fatty acids. There are saturated chains, there's unsaturated chains, and they have different kind of structures. If they are super tightly packed, well, then it's a bunch of saturated fatty acids. If it's not so tightly packed, you've got a mixture of saturated and unsaturated fats. Now, lipids have all these different classifications, and the last of which is your steroids. Now, I bring up steroids because they are important for digestion of fats. So, cholesterol, for example, is a molecule that consists of three six-membered rings and one five-membered ring. These are all fused together. And cholesterol is the most common steroid in animals and the precursor for lots of other steroids in animals. Here is your general structure for a steroid or for cholesterol. Now, this is significant because these one, two, three, four rings are kind of the bare bone structure. Bile salts or bile acids, if you look at it, they're pretty darn non-polar. Lots of carbons and hydrogens, and not a lot else. So yeah, with cholesterol, we've got a polar head group, but that's about it. Now, a bile acid can act as a detergent. A bile acid is a derivative of cholesterol. It is synthesized in the liver, stored in the gallbladder. You have these different places where you can modify them to make different bile acids. Do you have to know these names for these bile acids? Am I expecting that? No, I'm not. What I want you to understand is the sequence of events. Dietary fat goes into your stomach. That's going to trigger the release of bile salts from your gallbladder. Now, can anyone tell me what's a detergent do to, I don't know, an oil? What does a detergent do? When you think about detergents, I always think about soaps. And I think about rubbing my hands together. What am I trying to do with a detergent in my hands, rubbing them together? Oh, Dylan, nice. Emulsifies and disperses. I love it. I was just going to go with, maybe a little bit less, I'll say classy or technical, less technical terminology. I was going to say I'm using it to try to break things up. I'm trying to break up, dirt. I'm trying to emulsify non-polar substances, make them a little bit more liquefied. Now, that's exactly what these bile acids that are stored in your gallbladder are going to do whenever they hit your stomach and hit that dietary fat that's in your stomach. They are going to help break up that dietary fat into large lipid droplets and then subsequently into smaller lipid droplets. Okay, so what we're intending for is these bile acids to help disrupt and break down dietary fat into smaller pieces. So think about it if you're playing along at home and you're like, what's my takeaway? Your takeaway is, I got to get pen. Your takeaway is bile acids, BA. Break fats into smaller chunks. Because then it's a little bit easier to process. It's a little bit easier to do something with. And whenever we have that, we can bring in lipases. Okay, so we got fat bile acids, lipases. Lipases are the enzymes that take, sorry, lipases are the enzymes that take a triacylglycerol and break the individual fatty acids off. So think about it as fats, dietary fats, is lots of TAGs, triacylglycerols, also known as triglycerides. Bile acids, well, they're going to help break those up a little bit, but you're still dealing with maybe smaller TAGs. Lipases will take TAGs and break them into FAs. Could you give me a 1 if that, okay, I follow that, and a 5 if, wait, I missed a step. Okay. Now, a fair question is, well, a lipase is an enzyme made up of what is a lipase made up of, do you think? Probably amino acids. Well, a lipase is an example of a protein. Proteins are made up of amino acids, so yeah, I'll take that. But I was looking for amino acids. Amino acids are typically going to be, well, they're going to like to be soluble in an aqueous environment. Not every amino acid, but lots of amino acids are polar, polar uncharged, polar negatively charged, polar positively charged. Now, how does an enzyme, something that likes to be in an aqueous environment, sorry, I just had a moment that I was scared I was sharing like not what it was on the screen, on my screen. How is that aqueous substance? How are those amino acids going to interact with something that, well, a lipid? It's, it's largely non-polar, it's largely hydrophobic. It's not going to be soluble. How is that going to work? Because enzymes are generally soluble proteins. We need to be able to reconcile that. And the way that intestinal lipases, these are lipases that are found in the intestines, the way that they work is they have a region within the protein structure that I'm going to circle right here. It's this yellow, it's simply called a lid. It's a structural component that will open up to expose a bunch of hydrophobic active site residues. Now, could anyone tell me the one letter code for an amino acid that might be found here, that might be an active site or a hydrophobic active site residue? Tell me the single letter code for basically a hydrophobic or a non-polar amino acid. Yeah, alanine's a good one, proline's a good one. Those will both work. Great. Leucine, isoleucine, anything like that would work. So in other lipases, you might have a channel that leads to an active site, but many of them have this lid structure where it just sort of opens up to expose that hydrophobic or nonpolar region. Now, this is all activated through something known as interfacial activation. When you have this lipid water interface, the protein is sensitive enough to basically, it's almost like the protein is on the surface of the water and it's able to tell, oh, okay, that's something hydrophobic right above it. If it's, 20 angstroms away from it, it's not going to do anything. But it's sensitive enough that whenever it gets right there, it says, aha, we're at the lipid water interface. So I'm going to open up. Now, this shows the remaining process where we basically take these micelles with the help of bile salts, break them down into individual components, and resynthesize them into smaller droplets, which can then be trafficked to different parts of the body. Now, if you go on to take metabolism, you will take this whole snapshot right here and you'll conquer it all. I know you will. For this class, what I want you to know is dietary fat, big chunk. They will go to the stomach. I'm going to erase one. I'm going to write this kind of as one complete or one stream of consciousness statement. Go to the stomach. BA or bile acids synthesized in liver, stored in GB or your gallbladder, break down into lipid droplets, then then into my cells. Lipases act on my cells, on TAGs of my cells. That's your kind of crash course summary of basically how dietary fat is ingested. And how we get to, well, a lipase is going to act on a triacylglycerol, which is going to get us the FA or the fatty acids that we want. Could you give me a one if you follow that process and the depth that I want you to know, and a letter if, wait, I'm mixed up, huh? I've missed something. Okay. 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 So lots of the information that's shown on this is not something that you're expected to know for this class. If you want to take another look at it and see different components like VLDLs, IDLs, LDL, or HDLs, maybe some of these terms that you've heard about with like lipid panels and a annual physical exam, by all means, this might give you some more information. What's the point of the previous slide about interfacial activation lipid water interface? Is that explaining the context of the lid? It absolutely is. Thank you for asking. That's a great question. Essentially, what this protein is able to do, I'm going to go back a little bit to here. So this right here is a micelle. So that is um, a micelle that a phospho or a lipase is basically able to go up right next to it. And what this is able to do is detect the where it is. Let's see, let me rephrase this. Um, the enzyme is sensitive enough. Okay. I'm going to use this top figure. If you look at the space right here, this structure right here, this protein right here that I just drew the star by, I want you to look at that lid, that yellow ribbon structure there. Could you give me a one in the chat if you see that? Okay, so that is the lid whenever it is closed. So the hydrophobic amino acids that are found in the active site of that lipase, those are not exposed to the environment. The reason for that is this box that I drew that's red, well, all of that is basically water or the cytosolic solution. There's, could you give me a five in the chat if you follow that? Okay. Now I'm going to erase this box. And now I'm going to scribble over this. I'm going to put a star right here. Now, the lid has opened up, exposing the hydrophobic amino acids within the active site. The reason for that is Basically, there is no water or space between the protein and the head groups of the micelle. So the point is that this enzyme right here is sensitive enough to detect what is basically right next to it, and whatever that thing right next to it is water or it is a micelle. Could you give me a 9 in the chat if that makes sense, or a Q in the chat if you still have a question or confusion about that? Okay. So the point is that the enzyme is sensitive. It can say, oh, well, there's water next to me. I'm going to keep the lid closed. And then it's like, oh, wait, that's a micelle. That's something I can kind of wrap my mind around. So lid, open up, hydrophobic residues exposed, process the micelle. Did that help relay the significance? Okay, great. I'm happy to hear that. So that interfacial activation, think about that as just a very it's an appropriate term for the lipid water interface. Whenever there is a lipid water interface, the enzyme gets activated by opening the lid. Okay, so this is kind of your rundown of everything that's happening. Now, what we want to do is we want to look at, well, what's happening? How is this, how is any of this happening? And there are lipases that are hormone sensitive. And this is an example of a hormone sensitive lipase. So, in order for a lipase to be responsive to a hormone, that hormone will not enter the cell. Instead, it will be perceived by a receptor protein. This is kind of a preliminary look at biosignaling. That receptor protein will activate an enzyme known as adenylyl cyclase. And the important takeaway for this is that it will make a molecule known as CAMP. Could anyone add to the chat, what do you think the C stands for? I'll give you a hint. We've already used this term. Cyclic adenosine monophosphate. Cyclic adenosine monophosphate, also known as CAMP, also known as CAMP, is what's known as a secondary messenger. Your primary messenger would be a hormone. That hormone is going to stimulate the production of cyclic AMP. Now, this cyclic AMP, as a secondary messenger, activates a protein known as pKa. Has anyone ever heard of pKa? If you have, add a 7 to the chat. If you've not, add a letter to the chat. Okay, so pKa is one of a series of different protein kinases. And what this protein loves to do is it's almost like an aggregator. It's almost like it receives stimuli and then it spreads it out. It's like it amplifies the signal. So when cyclic AMP is activated, or sorry, when cyclic AMP's concentration goes up, well, that could mean lots of different things. Essentially, what this means right here is that, hey, we've got reason to break down um, micelles to get a bunch of energy from it. So this cyclic AMP production is going to lead to the activation of protein kinase A. Protein kinase A is going to activate hormone-sensitive lipase. It activates it. I want you to look at this number 3 here. It activates it by phosphorylating it, transferring a phosphate group from ATP onto hormone-sensitive lipase. That X that I've just drawn shows the phosphorylated hormone-sensitive lipase. So hormone-sensitive lipase, when it's phosphorylated, is active. And what it will do is it will take a glycerophospholipid, or sorry, it will take triacylglycerols and split them into individual fatty acids. Now, those individual fatty acids will enter the bloodstream, be transported into an adjacent cell, a myocyte, a muscle cell, so that those individual fatty acids can be broken down through a process known as beta oxidation to create a bunch of ATP, CO2. Okay, so your takeaway for this slide right here is that CAMP can B, an activator, activator, vator of TAG breakdown into fatty acids. Now, yeah, that's the important takeaway from this. Then to say, well, in those fat cells, those adipocytes, that's where there's lots of triacylglycerols that are then broken down into fatty acids. Those fatty acids, FA, transported, I'll say exported to other cells for breakdown. So the kind of important part about this slide is found on the left-hand side. Um, Cyclic AMP, your secondary messenger, activates PKA, which activates hormone-sensitive lipase, which gets us these fatty acids. Those fatty acids are transported so that they can be broken down in other cells to get energy from those. Now, with that in mind, what's kind of neglected here in this slide is we've got a glycerophosphate, or we've got a triacylglycerol. Triacyl, or your three fatty acids, glycerol. Triacylglycerol. The three carbons of glycerol, what happens to those? Well, the glycerol portion of a fatty acid, or a triacylglycerol, goes to the liver. Interesting. Oh, wait, stop, press. The glycerol portion of a triacylglycerol goes to the liver. In the liver, that glycerol gets phosphorylated to make 3-phosphoglycerol. 3-phosphoglycerol reacts with FAD to produce dihydroxyacetone phosphate. Huh. Where have we heard of dihydroxyacetone phosphate? I'll give you a hint. It's a metabolic pathway that involves glucose. It starts with a G. Glycolysis. Does anyone remember what is the reaction where we take 6 carbons and split it into 2 3-carbon molecules within glycolysis? What's the enzyme that does that? My hint is that it starts with an A. Not AC. I'll give you another hint. AL. D. O. Lase. Aldolase. Our aldolase enzyme takes the 6 carbons from fructose 2,6 bis or fructose 1,6 bisphosphate and splits it into dihydroxyacetone phosphate and glyceraldehyde 3-phosphate. Could you give me a 1 if you remember that? But yeah, it's been a minute. Or 9 if I have never heard that before in my life. And you, sir, are a liar. Okay, seeing ones. That's good. Okay, so what I want you to see with this, though, is that this reaction gets us some FADH2. We talked about FADH2 in the tricarboxylic acid reaction. DHAP can then be used for making glucose in gluconeogenesis, which we haven't talked about, but that is kind of the reverse of glycolysis. Reverse glycolysis. This FADH2, oh, that can go into the electron transport chain. This is an E carrier. So the point is that that glycerol that we get from that triacylglycerol, we can use that to make more ATP ultimately. Now, in order to, okay, so now we're getting to individual fatty acids. Okay, so we've got our fatty acid. Maybe let's go with a C16 colon 0. 16 carbons. We want to figure out what we can do with that 16 carbon molecule, which, I mean, basically what we're looking at is a molecule that looks like this. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16. Okay, so we've got this 16 carbon long molecule. What we want to do is we want to take this end, those 16 carbons, and do something with it. The way that we do that is with an enzyme known as acyl-CoA synthetase. So that enzyme will take this molecule and it will take this molecule known as acetyl-CoA and take this right here and plug it in right there. Could you give me a 5 in the chat if you understand that we're taking that end of it and plugging it into this? Okay. Now, in the process of this, we have to use ATP, and we'll get some AMP and PPI out of it. Thank you for the correction with the 5. I was curious though. So we've got to plug this alkyl end onto this acetyl-CoA or this coenzyme A. CoA is what it's called. Now, what we want to do is once we have that molecule, which is this R represents the remaining 15 carbons. So our C16 colon 0, that is this thing right here. Could you give me a W in the chat if you understand that? Okay. This S-CoA that I'm underlining right there represents this whole thing right here. Can you give me an X in the chat if you understand that? Okay. Now, we have to use an enzyme. So this has all happened. These fatty acids are in the cytosol. Where we are going to break those down is in the mitochondria. So could we move this whole molecule? Do you think that we could move this whole thing right here? Do you think that this, let me rephrase that. Do you think this is smaller than about 75 amino acids? Give me a Y or an N to the chat. 75 amino acids. 75 amino acids, that would be, 200 some odd nitrogens and carbons at least. Just thinking about the backbone. So Y or N, do you think that this whole thing that I've got circled here is smaller than 75 amino acids? Yeah. So do you think that that could move across our outer membrane? Probably. Probably not too much of an issue. But then the inner membrane, that's the super selective one. We've got to get that from one side, the intermembrane space, into the mitochondria. How are we going to do that? We are going to do that with an enzyme known as a carnitine carrier protein. Now, this word right here, carnitine, has anyone ever heard that term before? Carnitine, if you've ever drank like a Monster Energy drink, maybe a Red Bull, or any sort of like so-called fat burner drinks, chances are it's got carnitine in it. Now, carnitine is a compound that, well, it is involved in transport of fatty acids and therefore involved in the breakdown of fatty acids. So reasons to say, oh, well, if I increase the amount of carnitine in my body, well, then maybe I'm going to burn more fat. Or at least that's what the marketing team agrees on. Carnitine is an amino acid. It's a non-proteinogenic amino acid. More than anything else, it is involved in the breakdown of fats because it's going to take our 16 carbon long fat and this enzyme known as carnitine palmitoyl transferase 1, which is a cytosolic enzyme, is going to take our fatty acid chain and attach it to this enzyme carnitine. The carnitine carrier protein will take anything that carnitine is bound to and move it from one side of the membrane to the other side of the membrane. If rather than a 16 carbon long fatty acid, we had a Ford F-150 attached to carnitine, what do you think the carnitine carrier protein is going to do with that Ford F-150 attached to carnitine? In my magical world of wonder, it's going to move it to the inner side of the membrane because the carnitine carrier protein looks for carnitine and says, you're moving to the other side of the membrane. If it has a Ford F-150, it's probably not going to fit in the cell, but nevertheless, we've got our 16 carbon long fatty acid connected to carnitine that is moved to the inner side of the mitochondria. Now that 16 carbon long fatty acid that is attached to carnitine, well, it's going to be released from carnitine whenever it gets into the inside of the membrane by an enzyme known as carnitine palmitoyl transferase 2. Okay, so think about this as, I always think about it as a sequence. CPT 1 helps you move or helps you attach to carnitine. CPT 2 helps you detach your 16 carbon long fatty acid from carnitine and put it back onto this S-CoA, that acetyl-CoA. Okay, so what we have on this side of the cytosolic side of the membrane is almost identical to what we have on the inside portion of the membrane. But how did we do that? Well, that means that we've got S-CoA on each side of the membrane. We don't have a substance, or we don't have an enzyme that can move that from one side of the membrane to the next. Now, what we have been doing all of this for is this right here. This is beta oxidation. Now, I think that we'll be able to get through the king's ransom of all this because what's taking place is here's our S-CoA, which is this whole thing right here. Think about S-CoA as the holder of your fatty acid. So we've got our S-CoA, and then here's our 16 carbon long fatty acid. Now, what we are going to do in all of this is we are going to go through these steps and we are going to clip off 2 carbons at a time. So in one round of beta oxidation is what fatty acid degradation is known as. You will remove 2 carbons. So tell me this in the chat. How many rounds of beta oxidation are required to remove 6 carbons? 3. Okay, so 1 round you'll remove 2. A second round you'll remove 2 more. And then a third round you'll remove 6. You'll get to 6 have been removed. Okay, so with that in mind, I want to ask you the question, and we'll work on this together. How many rounds will total breakdown a 16 carbon FA or fatty acid require? Now, let's think about this. Okay, so round one, we're going to take 16 carbons and we're going to end up with a 14C plus this molecule right here, acetyl-CoA. I'm going to say, I'll just label that as, yeah, A-CoA. Okay, round two, 14 carbons down to 12 carbons plus another A-CoA. 12 carbons down to 10 carbons plus A-CoA. Our fourth round is going to go 10 carbons down to 8 plus A-CoA. Our fifth round is going to be 8 carbons down to A-CoA plus 6 carbons. Sorry for switching that around. Our sixth round will give us a 6-carbon molecule that's broken down to 4 carbons plus an A-CoA. Could everyone give me a 1 in the chat if you follow what I'm saying by the A-CoA? A 2-carbon. Yeah, great. So then round 7, we're going to take a 4-carbon molecule. And could somebody tell me, I'm going to give you guys the chance to answer this. What are your products going to be of that? Got to vote for a 2-carbon molecule plus A-CoA. Okay. Now, I didn't emphasize this, but now I want you to take another look at this. When we took our first 16-carbon long molecule and we ended right here at the end of this pathway, we ended with our A-CoA, and then here's our 14-carbon molecule. That 14-carbon molecule, is that attached to that CoA? It is. So what do we call 2 carbons attached to that CoA? We call that acetyl-CoA. Could everyone give me a 5 in the chat if you understand that? Okay, so when we have something like this 7th round, rather than saying we break it down into a 2-carbon molecule and A-CoA, we can just say that we break it down into 2 A-CoA molecules. Could you give me a 9 in the chat if you follow that, or an E in the chat if, wait, I missed it. Okay. I'm going to go ahead and stop there. We'll pick this back up on Thursday, and we'll talk about biosignaling for a little bit. We already kind of had a little primer for that with respect to hormone-sensitive lipases, but we'll get more into this stuff and look at some more numbers and calculations related to beta oxidation on Thursday. But thank you guys very much for coming. I'm going to stop my recording. If you've got questions and want to hang around and have them answered, you're more